Virus infections always form a real threat and can spread very quickly. A recent example is the outbreak of tomato brown rugose fruit virus in tomato. In just a few years, the virus has spread worldwide. Keeping your crop virus-free is obviously the best method, but how to fight a virus once you have an infection in your crop? Viruses are not really alive, and therefore you cannot kill them using pesticides. You can only try to inactivate them, so they can no longer infect plants. Some viruses, however, are so strong that it's very hard to destroy them, and a pressure cleaner will not do the trick. Tomato brown rugose fruit virus is such an exceptionally strong virus. You need to heat it for more than 10 minutes at 90 degrees centigrade to inactivate it, so it can no longer infect plants, to kill it, so to say. It's so strong that on surfaces it can stay infectious for months. In addition to that, tomato brown rugose fruit virus, just like other tobacco viruses, is present in very high concentration, often tens of millions of virus particles, while only a few hundred of those particles are needed to infect a plant. Currently available laboratory methods, such as ELISA or Tachman PCR, are very sensitive methods to detect viruses, but they are unable to say whether these viruses are still infectious or not. So how do you know if a virus is dead or alive? So far, this can only be done through a so-called bioassay. In such a bioassay, you use plants to test if viruses are still infectious and can cause symptoms on a plant. However, for a reliable test, you need a lot of time and a lot of plants, sometimes even thousands. This is not only expensive, but also not very practical. Within the VIRTIGATION project, Wageningen University Research has developed a laboratory test through which we can determine if a virus is either dead or alive. This laboratory test can replace the bioassay and will give you an answer already within 1.5 days. The test we have developed that can distinguish between live and dead virus is also based on the detection of the genetic material of a virus, just like a Tachman assay. But in contrast to a Tachman, our test can show if the genetic material of the virus is still intact or if it's degraded. Because if the genetic material of the virus is degraded, the virus is no longer alive, so no longer infectious. In experiments, we have heated plants up infected with tomato mosaic virus, a virus which is very closely related to tomato brown rugose fruit virus, at different temperatures. These experiments have shown that even 20 minutes at 65 degrees centigrade is not enough to infect the virus. Only after 20 minutes, 95 degrees centigrade, the virus was dead and it could no longer infect the plant and cause symptoms. And this was 100% in line with the results we had already obtained from our laboratory test. So, to close off, we have developed a test that can replace an expensive and laborious bioassay to determine if a virus is still infectious or whether it's dead. This test can be used for many different viruses and can help to limit or even prevent the unwanted and often unnoticed spread of plant viruses.